All right. I just started, yes. Look, yeah, I would. Thank you. Yeah, I'm going to be so freaking paranoid now. Okay. This week we're covering senses, starting with general and then moving into specific senses. There are only like 80 slides. So, like, if we get done early, we get done early. If we don't, there's not as much to cover as usual. So, next week will be endocrine. Endocrine has like 130 slides. However, like if I go like 60 slides a day, that's 2 full lectures essentially. However, we already went through a lot of that. Okay, so I can just boom. Yep, we did that. If you have any questions, nope, move on. Okay. So let's start with reminding ourselves that we've already covered the central nervous system, right? I hate you, Heidi.
Sorry.
I don't really hate you, but damn it, I'm. It's good. Central nervous system. We talked about the brain. We talked about the spinal cord. We talked about spinal reflexes. And we did a little bit of motor. So what we are really focusing on is the sensory. So what this box here shows you is the central nervous system right here. We have peripheral nervous system coming in. That would be your sensory or afferent system going in. So I have this green box to represent everything sensory. All right. If the sensory modality is coming from the skin or skeletal muscles or any sense except for vision, it's somatic. If it's coming from your viscera, and by viscera I mean organs, it's autonomic. Okay? So associated with that, we have different types of neurons. Most of what you're going to see are unipolar neurons, but in like the eye and the ear, there are bipolar neurons there. Not really that important, just if it comes up, that's what's going to be there. And then just a quick side note, go through the central nervous system. We come out, that would be the motor or efferent nervous system. All right, we already talked about if you have motor neurons going to skeletal muscle, that's somatic. If it's going to smooth muscle or glands, then it's, of course, autonomic. All right, the way we classify sensations, somatic sensations, is according to basically what triggers them. All right, so mechanoreceptive sensations are stimulated by mechanical displacement. What's mechanical displacement? Did you feel that? All right, that's mechanical displacement. He felt it because I displaced his skin, which displaced a certain part of his sensory receptor, which allowed that to be interpreted in the central nervous system. So that would be an example of tactile, touch, pressure, things like that. And then there's proprioceptive. Proprioceptive is balance, right? Why I don't fall down. We have thermoreceptive. Those are stimulated by temperature changes. Why temperature changes? Because you adjust really quickly to a given temperature. But when that temperature changes, then you feel it, right? So it's not how it feels at the moment, it's how it feels when it's changing. Okay, and nociceptive. And of course, that would be pain. Okay. So general receptors, located all throughout our body, allow us to do and feel different things in different areas of the body. As far as this picture, I guess, of receptors goes, these would all be mechanoreceptors. Right? Notice they feel touch, deep pressure, light touch, stretching of the skin. Right? Things like that. This one right here and this one right here would also be thermoreceptors. Okay, so your Krauss, German, corpuscle is cold, and I did a little bit of extra reading. We don't really know what the warm receptors are, but we think they are associated with Ruffini's receptors. And then, of course, nociceptors up here are free nerve endings, and the two at the bottom we're not going to cover because we did that when we covered muscle. Those are your Golgi tendon organ and your spindle apparatus. Okay, so just examples of general receptors. The question that's going through your head is, do we have to know all those? We will cover them in detail in specific sections. Special sensory receptors outside of general. So you notice general is like mostly like touch kind of stuff, like general sense about the environment. Whereas special sensory receptors detect very specific things. So photoreceptors, for example, they detect light. What does that allow you to do? See. Chemoreceptors detect chemicals in the air. All right, that allows you to, or in liquid, I guess solids too. They allow you to taste and smell things. Okay, I saw a meme on Facebook the other day that said, if you smell poop, does that mean that poop particles got into your nose? The answer is no. It's the answer is hydrogen sulfide got into your nose from the bacteria, not necessarily poop particles. All right, baroreceptors detect blood pressure, and sound or vestibular receptors allow you to detect sound, hearing, allows you to hear. And vestibular equilibrium, what does that allow you to do? Balance. You want to do a good experiment with balance testing? The day after all your finals are over, go to the bar. Some of you are like, don't got to tell me twice. All right. So all of our sensories are what we call a sensory modality. So if you hear that term, sensory modality, we're talking about the modality of sight or whatever. How the sensation is perceived is determined by two factors. One, what are the characteristics of the receptor that's detecting whatever it is? Okay, how does that receptor work? How does it result in an action potential that gets sent to the nervous system? All right, so one is at the end organ, skin, eyes, ears. Okay, what are the characteristics of the receptors there? The second is, where does it go in the brain, and how does it get connected to other areas of the brain? Okay, because I'll put it to you, if you have every single pain receptor in your body located on your forearm, but that didn't go to your central nervous system, you wouldn't feel pain, right? So it's very, very dependent on where it makes the connections. All right. The fact that nerve fibers coming from these receptors only transmit, give you a stupid example, touching my skin only transmits information about touch, not sight or smell or hearing. Okay? And it only goes to one part of the brain related to that function. That's called the label line principle. All right? Every axon is dedicated towards a specific sensation going to a specific part of the brain. All right. So this is a slide kind of outlining the general characteristics of sensory receptors. We're going to go into more specifics as we move along. All right. Mechanical, does that say mechanical deformation? Again, Stephen, I'm gonna use you as an example. I'm touching him. I'm deforming his skin. That deformation of his skin deforms the receptor associated with that, and that deformation of the receptor opens up ion channels, which leads to depolarization. All right. So the next slide shows kind of how that works, because we're going to lead into general senses after that. Chemicals. Chemicals bind to receptors. When a chemical binds to receptors, let's say between... What's this? Synapse. Okay. When chemicals bind to receptors in a synapse, what happens? You open up sodium channels, and sodium goes in. So if chemicals associated with smelling bind to a receptor, they're going to open up channels and depolarize that neuron. Okay? And then electromagnetic radiation, or light, has a very specific way of opening receptors, which we'll talk about later on. It's a little more complicated. That's why we're not going into it now. Okay? The point is, all of them, no matter which one they are, we'll take a membrane and make it more permeable to ions. Now, if you've had physiology before, especially nervous system physiology, is every single ion channel we're going to open throughout any of these lectures, this lecture, going to be a sodium channel? No. Not everyone's going to be a sodium channel. Some are going to be potassium. Some might be calcium. Okay, so pay attention to those specific details when we get there. Okay, let's go back to touching Stephen. I'm not going to touch you again. Tell you a quick story. I think everybody in here knows I'm taking nursing classes. My very first class, you want to know what they had me do? Write a professional email. I was like, you have got to be shitting me. And then we had to put together a PowerPoint. Like, again, you've got to be shitting me. And in order to put together this PowerPoint, I had to watch these videos on how to make an effective presentation. Oh, my God. So I watched the videos, and I actually learned quite a bit. One of the things I learned is I'm really old. I learned how to use PowerPoint back in 1998. Okay? Things have changed since then. I learned that there's a function to pull pictures directly into PowerPoint without copying and pasting from the website. I was like, that's cool. So that little finger right there is an example of that. I was proud. Old dog learning new tricks. All right, so let's assume this is a touch receptor, like when I touch Stephen. We're going to make a movie about that, touching Stephen. I poke it, I displace the membrane. When I displace the membrane, I also displace the area of this axon, and that's going to open up channels. Okay? Specifically for general senses, it's going to open up sodium channels. All right? What's sodium going to do? Always, it's going to flow in. All right? And at that specific location, what happens to membrane potential if resting is minus 70? Minus 65, minus 66. Okay? And that's going to happen in this local area. All right? Nothing's going to happen unless we hit what? Threshold. All right? Nothing's going to happen unless we hit threshold. If we hit threshold, let's say threshold is minus 50, If we hit threshold right here, is anything going to happen? And it just stays right here. Is anything going to happen? No, why not? Do what? No. Well, no, so yeah, it only traveled this far.
But it's not going to send an impulse to
Why?
the brain because it's only that area that got depolarized.
Okay.
It's not in the deformed area?
You need a receptor potential.
Why do we care about threshold potential? What does that do?
That way you don't feel it.
Right here, we have voltage gated sodium channels, right? And a shitload of them. Okay? So these detect minus 50. When threshold reaches those voltage gated channels, then we send an action potential. Does that make sense? So that's what I was trying to get at. So you have to have a wide enough potential change that makes it to these voltage gated channels to initiate an action potential. Okay, then you'll send it. So if that doesn't happen, I don't send action potentials. And no matter how much I touch Steven, I'm going to have to use somebody else as an example. He's going to get it complex. Is he going to feel it? No, because I'm not sending the action potentials.
What do you mean he won't feel it?
If I don't send an action potential down the neuron that's going to his spinal cord and up to his brain, then I never send the signal from his skin to there. And therefore, he's not going to feel it. Okay? Now, the other thing with this is when I press on this, as long as I'm pressing on it, I'm sending action potentials. Okay? So if we look at that like on a graph, that's going to go, it's going to send action potentials. Okay? If I press harder, what's going to happen? They're going to become more frequent. If I get a little softer, they're going to become less frequent. So the intensity with which Steven feels the touch is related to the number of action potentials that are being sent. Okay? The more action potentials in a shorter period of time, the greater the intensity he's going to feel that touch as. Up to a point. Because you can only send so many action potentials at a time, and that's limited by the repolarization of your axon. All right? So eventually that will level out, and you can only feel so intense. So no matter how hard I press, he's not going to feel any more what word am I thinking of? Strength of that touch. Jesus. No matter how much harder I press, because I can't send action potentials any faster than the limit at which the axon can send action potentials. All right?
This may or may not be related, but is this kind of related to pain tolerances in people and why different people have like different pain tolerances?
That explains some of it, not all of it. Unfortunately, pain. I'm fascinated by pain, and I want to learn more about it because I don't know if you guys have learned about pain in this gate control theory. I think it's complete bullshit. It sounds like if you pay attention to citations, some of the stuff we're still teaching and some of the stuff we still know, we found out in the 60s. Like, why aren't we learning more? Like some of these theories, still from the 60s. Like, can't you do better? It's been, 60 years since then? Anyway. Get down off my soapbox. Yes, partially. What else was I going to say about this? But let's say I press a little harder, and Steven does feel an increase in intensity. What might be happening? Even though I'm sending the maximum amount of signals, rapid succession, why might he still feel more intensity up to a point?
You're activating different receptors
Good. You're activating more receptors. So if you go back, this can be tied to the same concept we talked about with muscles, as that recruitment. Okay, the summation versus the recruitment. All right. All this is trying to show you is that if, I'm going to do Maddie this time.
Sorry.
If I say I'm going to touch Maddie, it sounds even worse. I'm going to use Maddie as an example. There we go. All right. I touch her. You guys are crazy. It is so hot in here. Are you guys hot too? There is. Oh. I'm not in here that often. What was I doing? Maddie. There's a movie about that somewhere too. Touching Maddie. Touching Stephen, touching Maddie. Too many jokes. We'll leave that alone. All right, I'm touching her. And as I increase pressure, she will feel that. Sorry, that was a missed example. If you have low I'm struggling. No. If you have a stimulus, like touching, damn it, I knew I had an example for this. All right, go ahead.
Is it like you can tell the difference between 1 pound and 5 pounds, but 101 versus 105 kilos, all the same difference?
Yes, thank you. That's a really good example. The example I was going to use is if I have a flashlight and I put it on the lowest setting, I shine it in your face, and I make it a little brighter, you're going to tell it's a little brighter. If I make it a little brighter, you're going to tell it's a little brighter. If I make it a little brighter, you're going to tell it's a little brighter. But if I turn it up all the way to the brightest and shine it in your face, and you're like, and I'd make a little change in that, you're not going to see a difference. Okay? So at low levels of stimulation, you can get a wide range of being able to discern what the stimulus is. But at high levels, you can't tell that difference anymore. It takes a large change in the stimulus in order to be able to perceive that change. That's what I'm trying to say. So I think Maddie had a better example than I did. And according to the other Maddie, she should be teaching this class. I'm just messing with you. That was an inside joke with one of the groups. I don't know whose group that was, besides Maddie's. All right. We also know that our sensory receptors adapt. All right. Some of them adapt really fast. An example of that is Pacinian corpuscles, which detect pressure. And actually, a lot of your skin receptors adapt really quickly. A really good example of that is I put on my watch in the morning. Within 10 seconds of me putting on my watch, I don't notice it's there anymore. That's fast adaptation. Okay. Slow adaptation, and I think in your book it states that the longest adaptation they've found to date is 2 days. 2 days for that receptor to adapt. And we know that some receptors, like baroreceptors, they're finding out, don't adapt at all. They never adapt. That means, and this is going to sound paradoxical, but if you have a slow adapting receptor, that means you can respond to changes much faster. Okay, a slow adapting receptor will respond to changes much quicker than a fast adapting receptor. All right, so let's get into the Pacifics. You guys ever know somebody who says that? Like, specifically, I want to tell you. My son said that to me once. I about blew my lid on him. He never did it again. All right. So what the next few slides is a list of the different types of receptors that you can find in your skin, where you find them, and what they do. So some of them are going to be very familiar to you, like Meissner's corpuscles. I think everybody who's ever taken an A&P class knows what a Meissner's corpuscle is. It allows you to detect touch, light touch specifically. Okay, where do you find them? Fingertips, any place on your body, generally speaking, without hair. Skin without hair. Okay. They are what they detect: motion, grip control. Adaptation is rapid, like we just talked about. Use the watch example. I don't care that you know specifically what the receptive field is, but if you're looking at a bunch of receptors and I say which one has the smallest receptive field, you should be able to pick that out. Okay, so Meissner's corpuscles is 22. That's not the lowest. If you look at the next one, Merkel discs, those have the lowest. And I'm not really concerned with the type of fibers either. Okay. Now, if you look at the picture, and it's hard to see on that, where are Meissner's corpuscles located within the cross-section of the skin? Right? But within that, if you say superficial to deep, where are they? They're more superficial. Okay? And that makes sense, right? Because if you want something to detect light touch, you want it to be near the surface. All right, so those are going to be near the surface, which means it's not going to take much. So I will put it to you this way, Stephen. When I touch Stephen, right now I'm probably activating Meissner's corpuscles. But when I start pressing, then that's going to be more of Pacinian corpuscles. Okay, so I may be changing the modality. Merkel discs, think fingerprints. That's where they're going to be located, within the ridges on your fingerprints. So if you have receptors that are way towards the surface, located on the ridges of your fingertips, think texture. Okay, they will help you with detecting texture. Right? These are slow adapting because in order to detect changes, in order to detect texture, you have to be moving. It's got to be changing all the time. So they're going to be slow adapting so you can react to that. All right, receptive field 9, which means that is lower, right? So they are more highly concentrated. Pacinian corpuscles, notice that those are deeper, well within the dermal layer. Okay, so if they're deeper, they're not going to detect specific light touch. They're going to detect more larger deformations in your skin. So they detect either deep pressure, that's not up there, but they do detect deep pressure, or vibrations. Think when I get done with my weed whacking, and I'm holding that thing and it's vibrating in my hands and I take it off, that vibration sense I'm feeling was from Pacinian corpuscles. Okay? They are very rapid adaptation. Okay, so I don't feel it after a certain amount of time, especially if I'm listening to one of my podcasts. And their receptive field is pretty big, entire finger or hand, so like much more broad. All right, Ruffini corpuscle. Despite what it says on this, what else do we also say Ruffini corpuscles detect? Remember a couple slides ago. Somebody said it. What was it? Warmth. Okay, they detect warmth as well. They're in the dermis. Notice that they are a little bit deeper as well. As far as touch goes, they detect tangential force. So sliding my hand across the table, that's what they're going to detect, shearing forces. All right? So their stimulus is going to be skin stretched, because when I do that, my skin is stretching, and they're slow at adapting, so they will allow you to respond quicker. Right? 60. So 60 millimeters squared. And then finally, free nerve endings. All right, free nerve endings, the first thing you should think of when you think free nerve endings is pain. All right, so this is your nociception. They are located pretty much everywhere. All right, they respond to pain and to temperature, because what you're going to find in once we get to temperature is that when you get either too cold or too hot, they both activate free nerve endings. Okay, so it's detected as pain, not as the temperature change. They are slowly adapting, so that means you respond much more frequently to them. And there are two types, myelinated and unmyelinated. What's the difference between a myelinated axon and an unmyelinated axon, besides the fact that one has fat and one doesn't? Therefore, this one tastes better and this one tastes... Speed. Which one's faster? Myelinated. Good. All right, so the myelinated ones will send signals much faster, whereas the unmyelinated ones much more slowly. So that would be like sharp pain versus like a dull throbbing pain. Okay. Okay, so that's generally speaking. We talked about the receptors, different types of receptors. We talked about how they work, and we started touching people to demonstrate that. All right, now where do they go? We've sent an action potential. Where is it going to go? So there are multiple pathways, but the two pathways you need to be responsible for to transmit sensory information from general senses, from these receptors we talked about, is the anterolateral system. So when these action potentials fire and they go down the neurons, they go to the spinal cord. Do they go into the back of the spinal cord or the front of the spinal cord? Back. All sensory goes in the back. Okay. Anterolateral, what does antero mean? Front, lateral, to the side. So that tells you where the tracks for those are located within the spinal cord. Okay, the anterolateral system is here. Dorsal column, medial limniscal system. Dorsal column, that would be this area right here. Median limniscal is something up in the brainstem. Okay, two pathways that you need to know. And these are going to be important when, especially, I think it's next semester, when you do Clin Med and you do neuro, we'll talk about different spinal cord injuries and how when these pathways are affected, they can produce different kinds of outcomes. One of them is very weird. All right, first we'll do the dorsal the DCML. Let's just say it that, the DCML. I'll write that down so I don't forget it. All right. The type of information that this specific pathway sends is touch, vibration, everything on that list. You guys can read. All right? So think touch goes with this. So what receptors are involved with that? Meissner's, Merkel's discs, things like Ruffini, things like that. Okay? So when they get activated, they will send it up this specific pathway. All right? On the right, you see a finger touching a feather. Forget it's a feather. Just say it's somebody else's finger because it doesn't say tickle up there, and tickle is actually, there's semantics associated here. You're touching something. I have issue with the fact that. All right. So whether it's in the foot or the finger, notice that it goes in through the back of the spinal cord. Where does it go? Directly to the dorsal column. Okay? And it travels all the way up the dorsal column. Okay? Same with the finger. It goes in through the back, travels up the dorsal column until it gets to the medulla oblongata. Okay? There, it makes a synapse. Okay, so that is your first neuron from the receptor to the medulla, up the dorsal column. Then that second neuron is going to cross over, which we call decussation. Right. Then they're going to go up, okay, through the pons, through the midbrain, and then they're going to synapse within the thalamus. Okay, so that's your second neuron from the medial lemniscal in the medulla to the thalamus. Now, what does the thalamus do? Switchboard. So it says, hey, you need to go to this specific part of the brain. Right? This is a really good example of how it does that. So this is receiving information from the finger that you just touched something. So it's going to say, hey, you need, I'm losing space. You need to go to this part of the brain, right? Because that is where your motor sensory homunculus is located for the finger, right about here, okay? And then the information from the foot, let's say they're both being touched at the same time. You get a signal going here and a signal going about midbrain, right? For your sensory homunculus. That's where that information is going to be interpreted. All right? That's the third pathway. So 3 neurons located in this pathway. Where do they cross over? Medulla. They cross over in the medulla. So, as they're traveling up, have they crossed over yet? No. Why am I making a big deal out of this? It's important. That's something you will need to know, and I will test you on. The other reason why I'm making a big deal out of it is when we talk about the anterolateral system, it works differently. It works differently. Okay? So, the other thing you need to know about this is that in the dorsal column, which is this whole thing in blue right here, it's spatially organized. And what that means is that the upper limb structures are on the lateral portion, whereas the lower limb structures are more medial. So upper limb is lateral. They travel up laterally. Lower limb travels up medially. Okay? So I'm going to test you right now. If we're talking about this right here, and I say you have a nerve, sensory neuron traveling up the cervical, is this coming from the right side or the left side? Left side. Right? My left is coming from the left side. Why? Because it hasn't crossed over yet. Right. Contrast that to the anterolateral system. All right. The anterolateral system, here's why I have an issue with that feather. I don't see a feather here. Pisses me off. You know what probably happened is like they copied and pasted from the anterolateral and said, "Oh, we'll just use the feather," but like they forgot that anyway. So these the anterolateral system transmits much different information. So notice the difference in the information. We're going to focus on pain. Okay, so let's say it transmits pain. Okay, pain from the finger or pain from the foot. Same locations, and it also has 3 neurons. Okay, so let's just use the finger. We'll ignore the foot. That travels through the back of the spinal cord into the gray matter. Yeah, into the gray matter, and it's going to synapse. Okay, as soon as it goes in, it's going to synapse. Okay? When it synapses, the second neuron crosses over at the level that the neuron entered. All right, so it decussates at the level that the neuron entered. And then it travels up the anterolateral tract of the spinal cord. okay, until it gets to the thalamus. Then the thalamus says, hey, I'm getting nerve impulses from the finger. I'm going to send that to the appropriate homunculus, sensory cortex, and so we can perceive that as pain. Okay, so what's the big difference here between the DCML and the anterolateral?
Crosses over before the medulla.
Which one?
The anterolateral.
Anterolateral crosses over before the medulla, so information from the right side of the body is traveling up the left side of the spinal cord, whereas for the dorsal column, median meniscal, information from the right side of the body is traveling up the right side of the spinal cord. Okay, so they're traveling up opposite sides of the spinal cord. No, I'm not going to make a big deal out of that one. Okay. So now we go into dermatomes, which I know, based on talking about to you in anatomy, that you guys love. Because your biggest question is, what do we have to know about dermatomes? Okay, I don't care that you know the dermatomes in this class. What I care that you know in this class is what a dermatome is. Okay, and a dermatome is an area of the skin that's supplied by a specific nerve from a spinal root. Okay, so this area right here, T5, would be associated with the spinal root T5. Okay, and the clinical significance of this, which is what you need to know, is that if somebody, let's say we suspect they have a spinal cord injury or a spinal nerve injury, and we go to them and we poke the area that's associated with T5 and there's no feeling, we've just confirmed where that injury is located. What if we go up to T4, T3, T2, and at T1 we finally start feeling things? We found the border of where that injury is. And then we go down and we find that the lower border is L4. Okay? We've isolated where the injury is. So that's the importance of knowing these. Right? The other interesting thing is that viruses, such as that which causes chickenpox and comes back later, tend to hide in nerves that follow along these dermatomes. So that's why you see somebody who has shingles later on, you'll see it going in a very specific pattern around a rib, which is where these dermatomes are located. Does that make sense?
Is it the same herpes too then?
Yes. Anybody ever heard of Cheech and Chong?
Yes.
I forget which movie it was. You just reminded me of it, Adam. Where, no, not, he said herpes. And there's a scene where he goes to get a haircut, and this guy's talking about a hair piece. But he's foreign, and he has this thick accent, and he goes, "Your hair piece. You want a hair piece? I'll give you a hair piece." He's like, no, I don't want hair piece. "Oh, no, take my hair piece." That's funny. Where are we in time? It's 2 o'clock. You want to take a break? All right, sweet. Me too. Because I need water. And I'm still recording, FYI. All right. Excuse me. We've touched. We've sent signals up the spinal cord. The next spot we got to talk about is what happens in the brain. Okay? At the beginning, we talked about how senses are perceived. It depends on 2 things. One is receptor characteristics, and the other is
CNS.
how it's interpreted in the brain. All right, so this is how it's interpreted in the brain. All right. Important landmark. What's this? central sulcus, and just posterior to that we have our primary somatosensory cortex. And what this blue represents is, again, the where specific sensations are interpreted. Okay? But let's assume this is the left side of the brain. Well, let's not assume it is the left side of the brain. All of this would be associated with the right side, the right thigh, being interpreted there. Okay? Because at some point it crossed over. Okay. So let's look at this creepy little homunculus again. And we've already talked about this. It's essentially the same for the sensory as it is for the motor. They mirror each other. The only difference is in the center of the brain you have genitals. And I think I mentioned that you can feel genitals, but you can't move genitals because there's no muscle in there. So there's this map. Okay? What I want you to notice about this map in particular is that the bigger the picture, the more neural space is dedicated to that particular spot. All right? So looking at this, the lips take up a large amount of space in the sensory cortex. All right? What does that mean? That means that there are a lot of receptors dedicated towards feeling with the lips. Okay, so there's a lot more input going there. So the point, that's how you would interpret looking at this. We're not going to go into any specific details about that. If the trunk and the lower body have the least area of representation, that means you have less receptors for touch in those areas. Oh, is this another one? I missed one. Yep, I missed it. Okay, not only do you have the sensory cortex, but you have these association areas too. So I think at the top, areas 5 and 7, which are here and here, those are association areas that will take the sensory information coming to the sensory cortex and connect it with visual input and auditory input and smell or taste input to give you a big picture of what's going on. All right, so these association areas are putting together multiple sensory modalities to get a bigger picture of what it is in your environment. Example I used to give in undergrad, and I'm experiencing right now in my home, and I have mixed feelings about this. I have a daughter, and she is 10, and she watches YouTube videos all the time, and she's getting into skincare now, and girly stuff, and scents, and lip gloss, and all this other stuff, and I'm like, oh my God, my house smells like a strawberry field. So that's a prelude to this. Way back when, let's assume, and maybe some of you are experiencing this now, you meet somebody and you're like, oh my God, this person is so beautiful, right? And they come close to you and they have this smell. It smells like a strawberry field, and you're like, oh, you have good feelings associated with that. And then she touches you, and then you associate that smell with that touch. And then every time you smell later on that smell, you think about this person and the way they touched you. All that stuff is going on in those association areas. Okay? So now I've touched 3 people in the class. File a complaint. Does that make sense? So those association areas, I put a bigger picture of what maybe love could be or infatuation could be. So that would be like how we interpret our general environment. If we lose these association areas, 5 and 7, one of the things we lose the ability to do is recognize complex objects. Okay? Like I'm looking here at Cody's workstation, and he's got gum and a badge and keys and earphones and a wallet and phone, a book, iPad, and a cup, most of which he loses at some point or another within the PA area. If I lost these association areas, I wouldn't be able to pick any of those out from each other. Okay? There's one really interesting condition where when you lose these areas, you neglect your contralateral side. So let's say it happens on your right, you neglect your left side to the point where you're like, that's not my arm. And you don't even acknowledge it's your arm, and you can't use it because you don't know. And if it does move, you're like, I don't know why that moved. Who's moving that arm? Like, what are you doing? And you get freaked out by it. Like, this is a legit condition.
So, when like a doctor's office and an elderly person comes in and they ask you to draw a clock, if you neglect the half of the clock, is that the same type of neglect as the association areas?
That'd be an association area problem. Yes. There's other things that the clock thing does that you can see, but yes.
So were you saying with his objects that you wouldn't be able to distinguish those as like your own objects, or you wouldn't be able to just distinguish them like between each other, like the gum?
Between each other. Like you lose the ability, like I know what an iPhone feels like, I know what those headphones feel like by just looking at them, but like I lose the association between that, so I'm like, I have no way to connect these with how I experience things in my daily life.
You can physically see that they're like different objects, but you can't like
Right.
know that that's an iPhone or
Ish. Yes.
like they're not all gonna look like gum like that.
They'll look like something, but you can't discriminate what one is from another.
And you still have like all your sensory like intact when that happens, right? Like it's just damaged.
Yeah, your sensations are all intact, but your ability to interpret them properly is not. Okay? Another example, like let's say I pick up his keys. Like I try to make a phone call with him.
So like the function of the object, like the purpose.
Yeah, you're associating it with, like, you don't know that that doesn't feel like a phone. So even though you see it's a key, it feels like a phone. Weird things. Yes.
How does phantom sense work when like someone goes into the military and loses their arm or something and they feel as though it's constantly tense even though it's not there?
Good question. Say I cut off my arm. Okay? When I cut off my arm, I lose all of the receptors in my arm, right? You agree with that? Do I lose the pathways going up to my brain, though? No, so those pathways are still there, and those nerve endings, when they get irritated or randomly fire, they can create this sensation that those areas are still there, and that's what we refer to as phantom limb syndrome.
The association area be like turned off when you're sleeping. Is that why like when you hand someone who's like half asleep a remote and they'll like put it up there? You like say you have a phone call and you hand them a remote, they'll literally put it up there. You'd be like, hello, because they like are too asleep to notice that you didn't actually hand them.
That's a really good question. Probably. I don't know the answer to that, but that's a really good observation. I'm going to have to look that up.
Doesn't that simulate like dementia patients, like not knowing that recognition is like how you have a sleep?
Maybe not in that area specifically. Streaming.
Or just tired too.
What, you pick up a banana and try to use it as a phone?
No, I saw a video of a nurse that got off her shift and she was trying to put her key to her door like this, like it was her bag.
Oh.
Done that.
So,
You come home from
so
work
hit.
and you
I don't.
use your work badge to turn it on. You did 100% done that.
So yeah, what Steven said was like when you're super tired, you kind of those signals get mixed up and you use things for the purpose they're not supposed to be. used for. So really interesting stuff. So like, we have this brain map and we have all these areas. You guys are like, oh my God, I have to know those areas. But then when you mention what happens when they go wrong, you're like, oh, yeah, okay, 5 and 7, sweet, got that. Phantom limb. No, that's not phantom limb. Messed me up. All right, now we have to have sensory information coming into the brain. And that sensory information has to get processed. And by getting processed, I mean it goes to like hundreds, thousands of different interneurons in a circuit. So we're not going to talk about the specifics of how that works, but if you look at this picture, we've seen this before, right? This is how the motor cortex is also organized. So we have layers, 1 through 6. Same layers in the motor cortex that are in the sensory cortex, so this is nothing new. But what is different here is that I'm going to kind of explain what the different layers do for sensory information. So if we're talking about layers 1 and 2, this would be superficial, so more towards the outside of the gray matter, whereas this would be deep. Okay? And so layers 1 and 2 towards the surface, they're the ones that are receiving incoming sensory information. So that sensory information goes all the way up through all the layers, bypassing thousands of neurons to get to the layer 2 and 3. And they receive diffuse input from lower brain centers. So you get a bunch of information coming in, and it goes to this area. Okay, and this area says, all right, we got to figure out what to do with this. Layers 2 and 3, so notice the layers that are receiving input overlap with the layers that are sending information to closely associated areas to allow that diffuse input to be more finely tuned. So layers 2 and 3 are communicating with other neurons to fine-tune that diffuse incoming signal. Okay, so instead of feeling like I've got all this information coming into my hand, all right, this is just coming from the finger, and it's just light touch. All right, this is hard to see, so if you guys can't see it very clearly, mark it. It's these purple arrows right here. What's different about these purple arrows that's compared? That was her funny bone. That's different than the arrows you saw in the previous slide. They're going down, okay? But this is sensory information. Where does sensory information go? Up. So what layers 5 and 6 do is they're sending signals that came down from those upper layers to different parts of the brain that are lower than the sensory cortex. So brainstem, spinal cord, okay, thalamus, to go relay that information to something else. Okay, so that's further processing of the information. If you're going to the brainstem or the cerebellum, what kind of information is that probably? What kind of information is that probably? Yes, that's what I want to ask. If it's going to the cerebellum. Information about position, where you are, okay, proprioception, okay? So that's what layers 5 and 6 are doing. So all those layers are involved in sending the signal coming in and parsing it out to make sense of it. All right. Now let's take that a little bit further. All right. In this picture, we still have layers 1 through 6. Okay, going down. Okay, so this is more superficial. These layers are divided into columns, and each column represents a different structure. So if we have sensory information coming up to the sensory cortex, here, the information from the second finger is going to be going just to this column. Third finger, just to this column. Fourth finger, just to this column. Okay? And then, seriously? Within each column, that's subdivided. Yes, it's that problematic. Into different modalities. So, would you agree that your second finger would have information coming from Meissner's corpuscles, Merkel's discs, Pacinian corpuscles? So that would be subdivided within that column. Okay, so we say that information is just going up to these areas, but it is super organized, despite how that map of the layers looks. Okay, so I'm not going to test you on anything like that. Like there's no way I can ask a question about that besides having you write an essay, and we're not doing that. Okay, but I want you to get an appreciation for how organized this really is. All right, so that's where all the information goes, and that's kind of the anatomical pathways of how it gets processed. How do we interpret it now? Okay, if we have a single point on the skin that's touched, there's my finger again, on the skin, the pathway's going up this way. Okay, that's why I put the finger down there, because 2 years ago I taught this lecture, I didn't have the finger there, and I'm on autopilot at this time, and I was going down. And so that lecture got all fucked up. So you're going up. Now, what do you notice that's happening as you go up? It's branching. Okay? So you have one neuron sending a signal to many neurons. That is what we call a divergent network. All right? What do divergent networks allow you to do? Okay, spread the message to a wider field, potentially, and there's one more. Amplification. So you spread the signal across a bigger area, and you're able to amplify the signal. Okay, so, yeah. All right, now, let's say we have a situation where we have 2 points being stimulated on the same part of your skin. You guys ever did this in anatomy and physiology labs, a 2-point discrimination? Okay, your ability to discriminate 2 points as 2 separate points instead of 1. You have 2 separate points. Both are on diverging networks. Those diverging networks overlap. Okay, so you got, if what we're going to talk about doesn't happen, you have this stimulus curve, which says, hey, I just felt something touch me. However, there are neurons in this pathway, these pathways, that inhibit neighboring neurons. And if you inhibit neighboring neurons, they're not interpreting that signal. And so you cancel out some of this signal that's being sent in the middle, and you're able to discriminate those two as separate points. That's what we call lateral inhibition. Because of the overlap of these neurons and their ability to inhibit each other within that overlap, you can inhibit the signal to be able to discriminate 2 separate points from each other. And that's it for touch. Okay, the hope today is to get through thermal sensations, taste, and smell. And those there's not very much associated with those slides. What slide are we on anyway? 28. 28? Okay. Thermal sensations. We talked about the receptors, cold and warm. Where they're located everywhere, but cold receptors, highest on the lips, lowest on the trunk. I argue with that because I have no problem putting cold water up to my lips, but I will not jump into that cold pool. Okay, and we talked about freezing cold and burning being the same sensation because they activate pain receptors, not necessarily the cold receptors. Now, this is really interesting because cold receptors respond from 7 degrees Celsius to 44 degrees Celsius. And 44 degrees Celsius, does anybody know how much that is in Fahrenheit? About 120 degrees. And that's a cold receptor. Okay, so why do they call it a cold receptor and a hot receptor? Because cold receptors are colder than warm receptors, not that they're specifically cold receptors. Does that make sense? Okay, so cold receptors detect this range of temperature, whereas warm receptors detect this range of temperatures, and they're named according to one is higher than the other. Okay, mechanism of stimulation. How do these at the receptor level get stimulated? The answer is nobody really knows for sure, but they think it is a change in the metabolic rate of the receptor. So think about if you, this isn't, don't take this as Bible verse. If you have a cold, a change in temperature towards the cold, and your membrane receptors get colder and the channel opens slower, that would be an example of metabolic change in the cell. All right? That's not true, but that's the best example I can think of right now to tell you what this means. Okay? And then that changes the rate of intracellular reactions. And that's it for temperature. Okay? So the big takeaway there, the type of receptors and the range at which they respond. And where pain starts. All right, for smell. Oh, man, I was going to bring in a skull. Smell. Smell comes from what we call the olfactory membrane, coming from cranial nerve 1. Sweet. You guys could have gone cranial nerve 1. Maybe you did, and I just didn't pay attention.
No comment. I'm kidding.
So where are these located? If you remember from anatomy, in the skull, you take off the skull cap, and there's that crista galli sticking up. On either side of those are cribriform plates, and that's where your cranial nerve 1 kind of sits in. Okay, they sit right in there, and they have little appendages that go down through the little holes in that cribriform plate and into your nasal cavity. Okay, so that's what is, there's the cribriform plate right here. Here would be your crista galli, and then here are the little appendages coming down. Okay, so this is the bone, and these are the neurons that are coming down, and they're receptors within the epithelium. Okay, so that's where they're located. On these receptors within the epithelium, we have these cilia. Now, these are different type of cilia. They're not the kind that move things. They're the kind that detect chemicals. So they have chemical receptors on these cilia that detect chemicals that are in the air, what we call odorants. Okay? When a chemical binds to those receptors, we have a change inside the cell that sends a signal. So how does that happen? All right, so let's assume this is your cell. These are the cilia. And on the membrane of that cilia, we have this set of proteins. This is called a G protein coupled receptor setup. Okay? And if you remember from cell biology or molecular biology, whichever you took at whatever college you went to and what they call it, you have your chemical. Let's go with the poop example again. It binds to the receptor. It activates a G protein. G protein activates adenylate cyclase, which produces CAMP, which is known as a second messenger. And that will bind to sodium channels and open them and allow the sodium to come in to the cell. Okay? The only thing I don't like, well, no, never mind. Okay? And when sodium goes into the cell, what's going to happen to the cell? It's going to depolarize. Okay? And if it depolarizes enough within it'll send a signal down the axon. Okay? Now, in order to be an odorant, because we can't smell everything, it has to be volatile. That means it has to be able to float up in the air. So even if you go down and you sniff the table and you smell something, that means something on that table is getting into the air, even if it's at a very low level. Okay? Has to be volatile, has to be slightly water soluble to be able to get into the mucus, and it has to be slightly lipid soluble to interact with the membrane. So, amphipathic molecules. Right? Olfactory receptors adapt very slowly. Again, we talked about slow versus fast adaptation. But the sensation of the odorant adapts rather rapidly. So where does that occur? Brain. The sensation of it adapts rapidly. Okay, this is why, and we're sticking with the poop example. You're sitting in the bathroom, you've got the door closed, you forgot to turn on the fan. Like, at some point you don't smell anything anymore. But then your wife walks by and she's like, oh my God. Okay, because your brain adapted to that smell. Okay. I wanted to use something different than, you guys have had something like this before, right? What's the example that everybody uses? Perfume. You walk into a perfume store and you don't smell anymore. God, I'm so sick of that example. So you guys get the dirty version. All right? When we smell, where does that information go? All right? First of all, is it going to go to the spinal cord? No, it's a cranial nerve, so it's going to go to the brainstem. Okay? Actually, it's not even going to go to the brainstem. We have two pathways, it turns out. So here is our olfactory bulb and our cranial nerve 1. We have an old area known as the medial area. That's associated with our limbic system, specifically the amygdala. What else is the amygdala associated with? Fear. Okay? So for the medial pathway, that's what these things are, like salivation, lip smacking, drooling, things like that. When you smell something, like the pot roast, right? Kind of your raw emotional response to that smell. The lateral, or the new, and by new they mean newly discovered, those are located in the temporal lobes, which is not pictured there very well. And those are where smell discrimination is processed. Like, can you tell the difference between, I order coffee from some place called Bones Coffee. It's in Florida. So I forget where in Florida. And they're known for their different flavors of coffees, and they just came out with a Indiana Jones set. So three flavors. One is like toffee. One is toffee, not coffee. Caramel and coconut, and I forget the third flavor. But they have this thing on it where you squeeze the bag and you can smell it. And so if I take one of the flavors and go, and then I take the other one, and then the other one, my ability to discriminate between those three is what's happening in the temporal lobe. Or good versus bad. Okay? Now, I gave the example of the bathroom and my wife walking by. She went by and she associates that with a bad smell. Now, if you grew up in let's say an environment where foul smells like fecal matter or rotting was a pleasant association,
You grew up on a farm.
or you grew up on a farm, yeah, that great example. You would have a different association with these smells than I would, for example, because I'm a city boy. So when we say good versus bad is a relative term. Okay, it depends upon what associations you've already made. All right, last thing for today is gustation. All right, what's gustation a fancy term for? Taste. All right, gustatory. So we have a bunch of different types of receptors for a bunch of different types of things, and these are the different types of receptors that are there. All right, on our tongue. Am I going to ask you how many receptors there are for sodium? No. But there are multiple receptors for multiple different things. One of the things that is kind of annoying that's still taught in schools is that there are certain areas of your tongue that are dedicated towards certain tastes. That's been proven to be not true, but it's still in textbooks. So that's not a true thing. These receptors are kind of evenly distributed for the most part. Okay? Now, all of these receptors work together to give us 5 different general tastes on our palate. All right? And that's sour, salty, sweet, umami. And whenever I say, because I'll use this word at home, I'm like, I'm craving something umami. And my wife goes, umami. Like, really? And bitter. Okay? Now, all of these, are pretty self-explanatory. But in blue, you notice that there is a threshold for taste. So you have to have at least a 2 millimolar concentration to be able to taste sour things, or taste things as sour. Sweet, it has to be at least 20 millimolar, which is probably why they sugar the shit out of everything nowadays. Right? Bitter, what do you notice about the threshold for bitter?
Very small.
Very, very small. So it takes very, very little bitter to be able to trigger a taste. Why do you think that is the case?
Poisons in the wilderness are better.
Right. Poisons, things that will harm you, are bitter. So if you taste that, you're going to want to spit it out right away. Okay? So the threshold for bitter is very low, and that's a protective mechanism. All right? Where do we have taste buds? Really? All over our tongue. Okay? We have different types. Circumvallate, which are these ones up here. Foliate, here. Fungiform. What does fungiform mean? Like mushrooms? Fungi? Fungiform? They look like they're kind of a mushroom shape. Different types and where they are located. Specifically. Now, all of these have different, you can have different receptors within them, but their location, the type is dependent on location. The only difference is we have these filiform papilla right here that are like little hairs. And filiform papilla are the ones that are most numerous on your tongue. And have you ever taken your tongue and rubbed your finger on it one way and rub it the other way, like a carpet, and it changes like color? Okay, that's because of these filiform taste buds. Okay, because they do that, they're responsible for touch. So your sensation of touch on your tongue. Okay, something that I don't hear talked about very much is we also have extraglossal taste buds. So that means outside of the tongue. We have taste buds on areas other than our tongue. Tonsillar pillars, you guys should know what those are from anatomy. Palate, where's your palate? Right up there. Epiglottis and proximal esophagus. Okay, so you can actually taste things back there. You ever Anybody ever experienced that before? Like noticed when you taste it, like in the back of your throat? Spicy, yeah, for sure. Sweet too. Sometimes you can taste it for a long period of time, way in the back. Okay, that's because we have taste buds back there. I find that fascinating.
Two questions. One, why do animals have hair on their tongues? Does that help at all with taste at all, or is it more just for scraping off what they're eating?
Those spike things. Yeah, that's for grooming.
Okay.
Yeah.
And then, when you burn a taste bud, does it lose its function, or does it eventually regenerate?
I guess that depends on the degree of burning. I would say kind of compare that to if you would burn your skin. If you burn it like through multiple layers, you probably won't get that back. But if you just burn surface, you might be able to grow those epithelial cells back. Okay. As far as taste buds go, our tongue, within all of these papilla, have up to 10,000 taste buds. That's a lot, right? So that's what allows us to get these wide range of tastes. Oh, I talked about my wife and umami. Umami is kind of that savoriness flavor. Like if I'm craving something umami, I want something that's like has a rich flavor to it that's not sweet. Usually they're kind of salty too, because MSG triggers that, and MSG is a salt. Okay? All of these flavors cause depolarization of taste cells by different mechanisms. We're not going to get into those different mechanisms. Okay? Bitter tends to use calcium. All right, and we talked about the bitter taste sensation and how that works. So I'm not going to get into any more details about that. Oh, and the average lifespan of a taste bud is 10 days. So your taste buds turn over every 10 days. All right? Once those taste buds are activated, they get transmitted. All right? If it's the anterior 2/3 of the tongue, right? That's the facial nerve. They get transmitted through the facial nerve. All right? So think about which kind of, no, never mind. So anterior 2/3 of the tongue. The posterior third of the tongue goes through the glossopharyngeal nerve. So front of the tongue, facial, back of the tongue, glossopharyngeal. Right? Those are the sensory modalities. Trigeminal nerve, V3, allows you to feel the touch coming from those foliate receptors. All right? The posterior aspect, all those extraglossal taste buds through the vagus nerve. All right? So as you work backwards, you go from cranial nerve 7, 9, 10. So it goes in order, right? Set up in order. When these get sent, they get sent to the solitary tract of the medulla oblongata, and they go to the cortex. Specifically, what's this right here? Thalamus, good. Makes a connection and then goes into the area where taste is interpreted. The gustatory cortex is located right here. So it's in one of the rarely talked about lobes of the cortex, the insular lobe. So gustatory is within the insular lobe. And that's it. Do you guys want to start eye, or we're done? Keep going? Yeah? All right. I'm only going to get through the first few slides of this, though, because the discussion of the actual physiology of how sight works will take a while to get into. So let's do the basic functional anatomy of it. All right, there's your eye. In anatomy, you guys actually Thursday, should be really working on eye, ear, and that last of your structure list, especially in the models room. I just looked at your exam today, and those models are used. So they will appear on the exam. I don't know which eye model I'm using, but they're all the same. They all show the same stuff. In fact, if you notice, the key for those eyes is the same key for all of the eyes. So just FYI. All right. Here we want to talk more about function. So with our eyelid, which is our palpebra, the muscle that controls that is the levator palpebrae superioris. Okay? And when we contract that, it pulls our eyelid up. All right? General structure of the eye, conjunctiva. So that is the clear membrane cover of the eye. Anybody who wears contacts and has had dry eyes before, you ever tried to take out your contacts and accidentally pull on that? Fucking hurts. The conjunctiva is rich in blood vessels, which you don't notice unless you're tired, drunk, high, okay, or have some sort of, or pink eye, which blood vessels will dilate, okay? So that's the membrane covering the sclera. So the white part of your eye is the sclera. All right. Then, of course, we have eyelashes. What's the purpose of eyelashes? I have a story about that, too. Do what?
Stops debris from going in the eye.
Right, stops debris. There's a reflex that if something hits your eyelash, like, let's say, a fly, you blink and cause it to go away. Or, so, yes, it does have a protective mechanism. I told you about my daughter getting into girly stuff. She showed me something. I don't know what it was. She's like, that's for attaching these, and she has a wide array of eyelashes. And I'm like, you've got to be kidding me. And they're really long. And I'm like, you're going to look stupid.
Take lots of pictures.
Oh, my God. And then we also have lacrimal glands and ducts. So the only thing I want to mention about that specifically is that your lacrimal gland is located here on the lateral portions of our eyes, but our ducts for the lacrimal glands are located on the medial portions. So what happens is tears drain into your eye from, superior and lateral, and then you open and close your eyelids, and it then drains towards the middle. Okay, so this is why, and then it empties into the nasal cavity. So this is why when you're crying and producing a lot of extra tears, and you have a lot of extra drainage into those nasal cavities, you tend to get stuffy too, okay, or a runny nose. All right, I was talking to some people in anatomy the other day about these eye muscles, these extraocular eye muscles. We have 4 main ones that are attached at obvious locations to our eye. That's the superior rectus, inferior rectus, lateral and medial rectus. Okay. The lateral and medial, very intuitive. If you contract the lateral rectus, what's your eye going to do? If you contract the medial rectus, what's your eye going to do? Okay. Then we also have these weird muscles, all right, these oblique muscles. We have the superior oblique and the inferior oblique. Now, if we look at it, here's our superior oblique. This one is supplied by cranial nerve, 4, trochlear, okay, the trochlear nerve. The reason why it's called the trochlear nerve, is because the superior oblique, before it attaches to the eyeball, moves through this pulley, and that pulley is called a trochlea. Okay, so it moves through this pulley and attaches the eyeball, and when it pulls, it rotates the eyeball. Okay, so it rotates, doesn't move it like left, right, up, down. It rotates. Right? The inferior oblique attached to the origin and the medial side of the eye, the attachment to the lateral side of the eye. So when it contracts, it's going to rotate the other way. Okay? So superior oblique will rotate this way. Inferior oblique will rotate that way.
The superior oblique rotates medially. Is that what you're saying? Or can you use medial and lateral in your expression?
With those bad arrows. If I pull on this, I'm going to pull the eyeball this way. Okay? If I pull on this, the eyeball is going to go in this direction. So I guess that would be a medial rotation versus lateral rotation. This is a bad pic. Oh, it's right here. Jesus. Yes. I know how you guys feel not paying attention to pictures or words on a question. I'm kidding. I'm human. We're all human. I get it. Okay. So, be aware of which muscles are supplied by which cranial nerve. So cranial nerve 3 supplies most of them. All right. The 2 obliques are supplied by the cranial nerves 4 and our cranial. Jesus, never mind. It's up there. This is what I really want to talk about. Now, as your eye is sitting there looking forward, okay, your eyes are not aligned with the direction of the sockets. The sockets actually go out this way, like they're oriented out. Literally like this, okay? So your eyeball, if it's pointing straight according to the direction of the muscles and the eye socket, it'll be pointed laterally. So both eyes would be looking laterally at the same time. All right? That's important because if you want to test into these individual muscles, you have to put your eye in a certain position to test those muscles. So if I wanted to test, we'll put it this way. If I want to test my lateral rectus, what would I do? Look lateral, right? If I wanted to test my medial rectus, I would look medial, right? Those are easy. In order to test my superior rectus, I would have to move my eyeball this way and then up. So when you're testing that, where are you actually looking? That way. Does that make sense? Why that's that way? Okay, so you're putting it in line with the actual muscle and then contracting that muscle. So putting it in line makes it go this way. Testing the muscle, it goes up. So that's why here, when you look at it, to superior rectus, look laterally and upward. That's how you test that muscle, but not what the muscle does when it contracts for vision. Does that make sense? So we're talking about testing the muscle versus what the muscle does when it contracts. Okay? When it contracts, where does it go? This way. Because your eye is oriented more medially when you're looking forward. Does that make sense? Okay, so that's the difference between what the eye movements do versus how you test the eye movements. That's what the upper versus lower picture is. I put this in physiology. I also put this in your anatomy lectures too, because when you guys do physical exam next semester, this is a big part of your eye exam. I'm supposed to be prepping your farm quiz. It's done. I told you guys that. All right. Here's an eyeball. General structure. We talked about some of the structures on the outside. Here's how I want to go about this, though. When light comes in, okay, first it has to go through this thin membrane. What's that thin membrane called? Conjunctiva. Then it has to go through the cornea. Okay, you go through the cornea. All right, then you go through a liquid portion called the anterior chamber. All right, after that you go through this hole. What's this hole called?
Iris.
No. Pupil, which is controlled by the iris. All right, and then it hits this thing. This thing is the lens. Once it hits a lens, then it shoots back through the majority of the eye chamber, the posterior chamber, which is filled with vitreous humor. Has anybody ever done an eyeball dissection? Yeah? What's inside the vitreous humor? Is it liquid?
Kind of jelly-like.
It's like jelly. Okay? The reason why it's jelly-like relates to its function. Its function is to make sure that and if you've done the eyeball dissection, you know what I'm talking about, to keep that retina pressed up against the back wall of the eye. That's its function. Okay, because this retina is not attached. It's not attached. It's held there by the vitreous humor. Okay, so when light comes in, it hits the retina. And the retina is where all of the light detecting mechanisms are located. All right, so that's what we're going to get into after today. Two other spots you need to know about the eye. One is at the very back, you have this optic disc, and the optic disc is connected to the optic nerve. So all of the retina converges onto this optic disc, and all those signals get sent down to the optic nerve. Now, because this is a nerve tract, and all the retina is converging on that, there are no actual light receptors located within the optic disc. That means that at some point in your vision, you have a blind spot. You guys, have you ever done that blind spot test? We'll do it next time I come in, but it's freaky. You have a piece of paper, and you have like an O and an X here, and you focus on one and move this and wait till the other one goes out of vision, and that'll tell you where your blind spot is. I'll bring in some cards. We'll do it next time. We'll do it Friday. Drink your coffee. Okay. The other spot you need to know is this fovea centralis, which is the very center, surrounded by the macula densa. Okay? That is where your vision is most acute. Okay? Where you get your highest resolution of images. Okay, right there. That's also where your color receptors are located. All right? Now, you guys... Oh, my. I forgot. Personal thing. Never mind. You guys are going to do eye exams next semester. In fact, you already did them when you were supposed to be doing lower limb, but... I can't let that go. I'm not mad. It's just funny. I'm like, why are you doing eye exams? When you look in the eye, you should be able to tell, you should see where the macula is and where the optic disc are. And what you're going to see in the eye is the optic disc is located medial to the fovea centralis, to the macula. Okay, so fovea, optic disc, okay, in each eye. We don't refer to it as medial and lateral, though. When we're talking about the eyes, we always refer to nasal versus temporal. Okay, nasal versus temporal. That's going to be a big distinction that you don't see anywhere else in physiology or anatomy. So, that's where we'll leave it. Friday we'll pick up with how the retina works, which is illuminating. I'm going to